Assalamualaikum warahmatullahi wabarakatuh. Welcome back to my YouTube channel, Aiman Story. Okay, today I want to share with all my subscribers with this new topic on Watson-Crick Model of DNA. So in the previous video, I already mentioned about the introduction to nucleic acids that consist of DNA and RNA, but for this video I only want to explain briefly about the model of DNA, suggested by James Watson and Francis Crick in 1950s, so very long ago. Very long time ago. So we have I will start with the brief explanation about the model. Model of DNA. So basically there are several scientists had involved in this discovery of DNA. So there was a race for DNA research at that time. So normally there are several names of notable scientists in that time. So one is James Watson, and then Francis Crick, and then Maurice Wilkins, and then last but not least, it is the only woman scientist involved in this DNA research at that time is Rosalind Franklin. Rosalind Franklin. So this kind of a DNA research had started based on the discovery of the DNA photograph. DNA photograph called Photo 51 that was discovered by Rosalind Franklin. Rosalind Franklin using DNA diffraction pattern. So basically this research was conducted using X-ray crystallography. So or other than that, we can call it as a X-ray diffraction experiment. So Rosalind Franklin managed to get the best photograph of DNA structure in the wet form. So the name of photograph is called Photo 51. This is a very really well known of the discovery on DNA research is what was related to this photograph, Photo 51. So following the model of DNA coined by James Watson and Francis Crick, so this kind of DNA structure, I will draw first, and then let's say this area, the left side, the left side, and this one is the right side. So it will start from 5' to 3'. So the direction is starting from 5' to 3', and then this one start 5' to 3'. So it will go upwards. So based on the model, James Watson and also Francis Crick deduce the structure of DNA is something like this. For the first point is that the backbone of the DNA structure, the polynucleotide chains, okay, the polynucleotide chains, there are two polynucleotide chains that are run in anti-parallel fashion. Okay, so this part of DNA change is run to this way, and then another DNA change run in the opposite direction. So normally the structure of DNA, there is a external part, the outer side, the outside structure of the DNA is related to the backbone. Okay, the backbone consists of sugar and phosphate. Okay, sugar and phosphate, and within the inner structure, the inside structure of the DNA is a new nitrogenous base. Okay, consists of 4 types: adenine, thymine, guanine, and cytosine. Okay, so the structure of DNA, okay, inside the nitrogenous base, okay, this arrangement of nitrogenous base is arranged stacking up on top of one another. So normally like this. Okay, I will draw. So let's say this one is A, and then this one is T, and then this one is G and C. Okay, so the arrangement of the nitrogenous base is the nitrogenous base will pair with the with its complementary base pairing. So that means that A is always pairing with the T, adenine is always pairing with the thymine, and then guanine always pair with the cytosine. So the interaction between the nitrogenous bases was linked with the hydrogen bond. So let's say for A and T pairing, there are two hydrogen bonds, and then for G, guanine, and cytosine, there are 3 hydrogen bonds. Okay, so in between of top and bottom nitrogenous base, okay, there is a space, okay, space in between. So the measurement is 3.4 Armstrong or 0.34 nanometer. So normally for the single DNA turn, okay, for single DNA turn, let's say like this, single DNA turn, okay, so like this one, single DNA turn. So normally for single DNA turn, there are at least 10 nitrogenous bases. Okay, so if one space of both nitrogenous base measured 0.34 nanometer, so if there are 10 bases, so the calculation is like this: 0.34 times 10, so 3.4 nanometer or 34 Armstrong. Okay, so other than that, the diameter of the DNA structure is diameter is 20 Armstrong, okay, or equal to 2 nanometer. Okay, so for the interaction of DNA structure to maintain the integrity of DNA, okay, we have to see that we have to see from the perspective of DNA location that DNA is located in the nucleus. So we know that the condition of nucleus inside the internal part of the nucleus is aqueous. Okay, aqueous because of there is a water inside. So the DNA structure, okay, the outer part of the DNA structure has phosphate group. So phosphate group. So there will be a negatively charged, okay, negatively charged, and then so the external structure of the DNA is polar or hydrophilic because of the condition of DNA, the external structure, the external surface of DNA is located in the aqueous environment or microenvironment in the nucleus. So in contrast, the internal structure of DNA, the model of DNA, the internal part is nitrogenous base, okay, nitrogenous base, along with the interaction of hydrogen bond that interact that bind the nitrogenous base pairing A and T, G and C. Okay, on top of that, there are other several interactions like van der Waals, okay, van der Waal interaction, and also hydrophobic interaction. So basically, in internal part of the DNA model, there is a hydrophobic interactions, and then for the nitrogenous bases, the stacking up arrangement of the nitrogenous bases was linked or was bind with the van der Waals interaction. Okay, so that's all from me today. So I hope it will be helpful for you for understanding on how the model of DNA looks like, and then see you in the next video on other topics on DNA or other related biology or molecular biology subjects. Okay, so please like, share, and subscribe my YouTube channel. Okay, thank you.